Welcome to my poster presentation. The goal of our work is to build a deep neural network which is less artificial than nowadays standard deep neural networks. This means without any derivatives of backpropagation inside. The idea is that when the network mechanisms are based on neuroscientific foundations, then the network should be capable to account for the circuit effects and the information processing of the brain without the limits of a strongly reduced circuit architecture. We focus on the visual cortex as the best investigated part of the neocortex and aim to be able to solve realistic tasks such as invariant object recognition. Moreover, we want to gain insights in the receptive field properties by advancing the knowledge on area V2. And finally, we want to observe the anatomical footprint of the learnings and link here the observed connectivity of the inhibitory neurons to its functioning, explaining why their connectivity is often described as unspecific despite learning is indeed specific. To do that, we had to combine three core principles which together enable these insights. These are synaptic, intrinsic, and structural plasticity. First, synaptic plasticity, to learn informative representations on the neuron level, where the excitatory plasticity extracts correlating patterns from the input, and the inhibitory plasticity causes response decorrelation by penalizing coactivity. Not to mention that this requires a form of synaptic homeostasis to bound the weight increase. We use plasticity formulations where the weight strength develop relative to the correlation of the pre- and postsynaptic activity. These rules have been demonstrated in earlier work to learn proper V1 simple and complex cell receptive fields. Second, intrinsic plasticity. The decorrelation of neurons through inhibition does not ensure that all information present in the input is also represented by a population of neurons. A further issue of the formulation of Hebbian learning is that synapses from more active neurons are more strengthened and can dominate the neural code. These imbalances in the representations of different patterns can harm the neural code in downstream layers. This issue can be resolved by aiming for equal mean and variance of all neurons within a population. Here, an example from the second layer of our model. With intrinsic plasticity, the mean activity of all neurons is in a narrow range, and all neurons are doing something. Whereas without intrinsic plasticity, a lot of neurons are doing nothing, while a few neurons dominate the neural code. And third, structural plasticity. It is difficult to decide on the individual connectivity, and statistical data are not very helpful to achieve good information processing. With structural plasticity, we can find the individual connectivity during the learning of the receptive field. This also allows to observe the anatomical footprint of the learning. Let's have a look on our model to get a clue how structural plasticity can be beneficial. We have 9 populations of excitatory and inhibitory neurons which are recurrently connected. These are some thousand neurons and about 1 million synapses. Dependent on the neuron type, the synapses are excitatory or inhibitory. For the initial connectivity, we use theoretical assumptions on the organization. Each neuron has a limited window of connected neurons, and the connections of the neurons are wet-and-top organized. This means neighboring neurons receive input from neighboring neurons. On population and layer level, the connectome was designed to follow findings on the neocortical connectivity. However, we do not know what is the right connectivity, for example, of these two neurons. Therefore, we refine during learning the connectivity structure with a mechanism we call experience-dependent structural plasticity. We base this mechanism on two basic neuroscientific findings. The formation of new synapses is more likely in the neighborhood of other strong synapses. We know the neighborhood because of our initial retentive organization in the model. And second, weak synapses are likely to be removed. Here we see the connections from LGN to a V1 layer 4 neuron with a typical simple cell receptive field. We see that in the surround of strong synapses, the dark squares, the formation probability for new synapses is high. So these are these red squares here. The removal is indicated by the blue boxes around the weight values. Weak weights, here white, which are typically at the border of a GABA-receptive field, have high removal probabilities indicated by the blue color. So the receptive field is growing and shrinking in its spatial surround. And the experience, which determines the weight strength, is the important factor which determines the connectivity. So what is the experience of our model? We train it on natural scenes. So not on the object recognition datasets we see on the next slide. Why this? Because it was found that when using natural scene images, then V1 receptive fields develop similar to the one found in macaque monkeys. So how good does the neural code represent objects? We tested a model on handwritten digits and it obtained about 98% recognition accuracy. However, this was achieved by the excitatory neurons of V1 layer 4. Features in deeper layers seem to be less suitable. So we also tested the model on the Coil 100 dataset, which was designed to test for invariant object recognition with real-world objects. What we found when training just on 4 object views, so the last case here, is that the features and the invariance properties of these neurons are beneficial and increase the recognition accuracy. Now, let's look on the receptive fields. We have already shown in earlier studies that our approach produces V1 simple and complex cell receptive fields. Thus, we look here for the properties of the neurons in area V2. They are poorly understood. A new theory says that the V2 neurons are more sensitive on naturalistic textures than to spectrally matched noise, while V1 neurons don't care. We found this trend also in our model. But we could advance the experimental data by fine-grained analysis for layers and neuron types. We found an increased modulation for V1 inhibitory neurons and even stronger for V2 inhibitory neurons. And as one could expect, their modulation increases gradually with layer depth. V1 generally shows a relative low sensitivity, but the highest layer, V2 layer 2-3, had much more sensitive neurons. Finally, we could analyze the learned connectome in our model. We found for the excitatory synapses, regardless of the connection, a clear trend that neurons which share higher response correlations also are more likely to be connected. This goes in line with mouse V1 layer 2-3 data. For the inhibitory connections, we see a different pattern. Uncorrelated neurons have a connection probability slightly below chance level, whereas negatively or positively correlated neurons are slightly above. However, the connection probability drops for highly correlated neurons. This is because of the function of inhibition to decorrelate the neurons. Only unconnected neurons can develop high correlations. When they are connected, then the correlation quickly decreases because of the increasing inhibitory weight. After a short time, these neurons are uncorrelated or negatively correlated, and the synaptic weight decreases. Weak synaptic weights are likely to be removed, however, structural plasticity is slow and neurons end up with a pool of seemingly unspecific synapses, despite inhibition acts highly specific. This is difficult to discover in experimental studies, as these strongly correlated neurons are rare and just exist in a relatively short time window. So experiments will just observe the statistics of the uncorrelated or slightly correlated neurons. Finally, some challenges of the computational modeling of the neocortex. A major issue is excitatory recurrence, which causes instability in the network. There's a trick to solve it by setting maximum activity, but this is implausible as neurons saturate at several hundred hertz. And moreover, it would harm the information representation of the system when driving neurons to saturation. This issue can be analyzed by the eigenvalues of the weights, which have to be below 1. But we do not know a solution which incorporates this into local Hebbian plasticity. Without this solution, we can also not useful model layer 5 and 6 because of their highly recurrent connectivity over the telomeres. The second challenge is to implement task-specific learning to obtain behavioral relevant representations. Political feedback can serve a mechanism for that by implementing something known as supervised Hebbian learning. That this can work has already been shown for the MNIST dataset. However, it is unclear how this can be a competitive mechanism to backpropagation. The advantage would be clear: a continuum between Hebbian unsupervised and supervised learning, which massively reduces the dependency on labeled data. Thank you for your attention, and I'm looking forward to the live session.